Do you want to?
No.
Okay. Well, welcome to the review session. We're gonna go over non-coding RNA. Here's a little meme for you. I don't really understand it, so you understand it. I understand.
Right.
Okay. So what is non-coding RNA? Pretty self-explanatory. It's just RNA molecules that do not code for proteins. They have a lot of different roles in the cell, and some of them are catalyzing chemical reactions and regulating gene expression. So some types of non-coding RNA. We have ribosomal RNA. So as we know, the ribosome is made up of RNA, more specifically ribosomal RNA. So the whole point of our RNA is to catalyze peptide bond formation in protein synthesis. So essentially, we'll get into tRNA in a second, but these tRNA are going to bring in amino acids into the ribosome, and the ribosome's job is to create a peptide bond between these proteins in order to create a chain. So tRNA transfer RNA, it's going to bring these amino acids over. It's going to bond complementary to the codon that codes for whatever amino acid it codes for. And then again, it's going to, the ribosome, which is made up of ribosomal RNA, is going to catalyze that peptide bond and create a chain. And it's going to link amino acids to the peptide chain. So some other non-coding RNA, we have small nuclear RNAs. They have the catalytic part of spliceosomes, so they help in splicing. Long non-coding RNA, these are mainly involved in the X chromosome inactivation. So in like female humans, they have 2 X chromosomes, and one's going to be condensed because they don't need 2 chromosomes that are coding for the same thing. I don't think you guys need to know that, but that's just what it does. We have CRISPR RNA. It's a common bacterial defense mechanism. These guide strands are going to target virus DNA, and they can cleave that DNA with the use of endonucleases. SRP is ribonucleoprotein used in protein translocation to the ER. And then we have RNases. It's a weird word. These are RNA nucleases that cut RNA. And the ribonucleoproteins are the RNA portion that is the catalytic part. So now moving on to RNA interference. Hold on. This is a mechanism of post-transcriptional control of mRNA. So this is after RNA is made and it's in the cell. RNA interference can help with suppressing that gene that's expressed. So there's RNA can be involved with RISC, which is RNA interference, RNA-induced silencing complex. There's also mechanisms with microRNA and short interfering RNA. So for RISC, which is RNA-induced silencing complex, this RNA will bind to RISC, and the RNA that is produced will be used as a guide strand to complementary base pair to a mRNA that has already been produced. So in a cell, if there's mRNA and say it needs to be degraded somehow, RISC will help create an mRNA strand or an RNA strand that is complementary to that mRNA strand that wants to be degraded. And after that RNA is produced, it's going to base pair with that mRNA. And RNA does not like to be double-stranded because it's very unstable. So once the cell knows that there's a double-stranded RNA, that RNA is going to be tagged for degradation. So that's a way how it can suppress gene expression in a cell. Does that make sense? And then the 2 different pathways are microRNA, small interfering RNA. And George will go into depth with both of those
Are there any questions?
Yeah, any questions? Either of those?
Is the RNAi, is that that binds risk and then the complex binds tags the mRNA it wants to degrade?
What's the glucosine? Yeah. I'm not really sure
Or
how
what was the
that
question?
works.
I was just basing it off the last slide. I was wondering,
This one.
is it RNAi is binding risk, or is this not, is RNAi not even just like an actual enzyme? I was wondering, is it RNAi that's binding risk and causing risk to target the mRNAs for degradation, or is that?
I think
Not know.
risk binds to that RNA. And then that is, since it's bound to risk, it like brings it to the mRNA that wants to be degraded. Is that correct?
Yeah, I think so. I'm pretty sure RNAi is just a name.
Yeah, RNAi is just RNA interference.
Yeah, just a name for it in general. It's not like a specific type of RNA or.
But risk is going to
Well-
bind to that RNA and it's going to bring it to the mRNA that wants to base pair, which will eventually get degraded.
Yeah, so risk will bind to the like interfering RNA and use that to
Mhm.
degrade. All right. Okay, so we're gonna go over the first pathway. It's microRNA and miRNA. So it's a post-transcriptional regulator, the guide strand for risk. And that's also true for siRNA. It regulates mRNA stability and localization. And it's very small. It's only 20-25 base pairs of double-stranded RNA. And it's endogenously expressed, which is important. That means it's only coded for in the genome and not like outside of the cell, which is that's siRNA. And it's not that prevalent in humans. And then we get into it more later, but I'm pretty sure for miRNA, this is the one where like it's usually not exact base pairing, while siRNA is. Yeah. And here's the pathway. So first, RNA polymerase transcribes the transcript. You can see in there. That's pri-mRNA. The graphic is wrong. This is pre-mRNA twice. I think it was long last semester too, but they never changed it, I guess. So yeah, pri-RNA or pri-mRNA goes through Joshua processing that turns into pre-mRNA, and it goes through the exportin pathway, which you guys learned last week, I think, or 2 weeks ago, and that brings back out into the cytoplasm, and then after that it goes through dicer, which turns into the mRNA duplexes, which you can see up there. Actually, I think that's the next slide. Yeah. Yeah, it turns into mRNA duplexes through dicer. And then now it's 20-25 base pairs long, and it goes to a risk protein complex, the duplex unwinding and incorporation to a risk complex. And then risk chooses one of those two strands to use as the one for as a guided strand, which you can then use to like base pair to the target mRNA that wants to degrade. So I think that's the next slide. Yeah. So I used the guide strand and I'm guessing the quiz yet for this week. Like the topic, yeah, topics. Yeah, when I took the class, there is a trick question on there where it's like true or false, is the like guide strand the exact same base pairs as the target strand, and it's false because it's complementary, it's not the exact same. So I remember that because that tripped up a lot of my friends back then. And then the end result usually is translational suppression or sequestered into p-bodies, which basically means that it's like irrelevant now. And then yeah, that's where it says mRNA in humans usually do not exactly base pair of the target mRNA. And that's important because that gives it more like versatility and what it can like base pair with and like affect. While like siRNA, well, if it's an exact match, it's also direct degradation, but usually it's siRNA that is like the exact match and degrades it. But yeah, speaking of siRNA, so yeah, here's a siRNA. A lot of similarities to miRNA, except that like it's exogenous, so it means it comes from outside of the cell. It's usually viral. So think about like, I don't know, maybe like a vaccine, it comes in from the outside rather than being produced from the inside. And it's usually an exact base pairing, like I said earlier. Next slide. So this time it starts in the cytoplasm. And I don't remember, or for my problem set, we had a question like this where like basically the siRNA and the miRNA pathways basically become like nearly identical once like they're both in the cytoplasm. So yeah, it's very similar with the mRNA where like it gets unwinded, RISC picks one of them to be the guide strand. Well, yeah, it's also with the help of a Go, which basically just binds into RISC. You don't really need to know that much about it, it does make it a bit more confusing, but yeah, basically it turns into a RISC a Go complex, which is what takes a guide strand and uses it to like target certain mRNAs. And so usually there's exact base pairing, and that turns into cleavage of mRNA, which means that it gets degraded. And also one siRNA guide strand can cleave many target mRNAs. That's also important to know. That was also a question on my problem set, I think. I don't know if it's on yours, but yeah. So for my problem set, it was like you're like pretend you're like in a research lab, and like what does this tell you about like guide strands? Yeah. And basically it tells you like one siRNA guide strand can target many. It's not just like one to one. That's important to know. As it's like reusable basically. Yeah, and here's just the differences between the two. So like I said, after it goes out, or after mRNA goes in out of the nucleus into the cytoplasm and the siRNA comes in, they're like very, very similar, like basically the exact same pathway, maybe up until like the very end. So yeah, that's really good to know. It makes it a lot easier to remember. Yeah, that's pretty much it. That's all the stuff we went over.
Could you just remind me what the P bodies are?
P-bodies. Honestly, I don't remember from when I took the class. I just know that like once the cell goes in there, it basically doesn't do anything.
Like, it's kind of just like I think it was like a jail.
Yeah,
Like, it kind of puts the RNA with the interfering RNA that's complementing, so it's double-stranded RNA. It's going to put it into a P body, which is
she's not gonna ask you a question like what is a P-body and describe it for
Yeah.
me. She's just gonna be like she's gonna want you to like know the function of it, which basically is the gel. Once it goes in there,
it's not going to come out.
Yeah.
It's either degraded or it just kind of lays dormant inside the P body.
yeah, basically whatever goes into there is irrelevant now, and you don't need to worry about it.
Mhm. Any other questions?
Nice.
Well, that's it for these slides.
Party cells say goodbye. Later, I have to split. Pretty funny.
So funny.
Do you guys record this lecture?
Yeah,
And the slides, like,
I think Tina posted them already.
Yeah, they're already posted.
Yeah.
I see that.
Last one, Monday or Tuesday, I think.
If you search up Tina Chen, she's the other TA that should have sent them out.
I wanted for,
We just used the second half of her slides.
Yeah,
I just have some problem set stuff.
yeah, that's all right. And we start recording or stop recording.